Hello, everyone. Thank you for giving me the chance present my neurodevelopmental work. Okay, cool. I just realized I'm the second person talk about something beyond synapses. So it's much easier. But I still want to tell you a small story that STXBP1 actually firstly identified from C. elegans. So that is a small worm. So loss of STXBP1 cause paralyze the small worm. So the synaptic theory, like STXBP1, control exocytosis in presynaptic terminal, perfectly explain the paralysis in this small worm. But in human patients, in those synaptic mutations, that theory cannot explain very well, because in those patients, the early onset epilepsy or autistic feature all remind us the neurodevelopmental defects. And also recently, people also find STXBP1 is also play a role, keep the neural survival, also play a role in endocytosis and vesicle trafficking. But what's the function of STXBP1 in neurodevelopment? Oh, sorry. So to solve that problem, we ask the first question: is STXBP1 is expressed during neurodevelopment? Basically, we just use immunohistochemistry. We use STXBP1 antibody to stain to detect the endogenous STXBP1 expression. So to prove that STXBP1 antibody is specific targeting the endogenous STXBP1, we generate STXBP1 knockout as our negative control. So in the STXBP1 knockout cell, we should not see any staining signal outside. We should be able to stain. We should be able to see signal. So this is the top panel showed our strategy. We use Cree knockout as STXBP1. At the same time, we use Cree induce the GLP expression. So GLP expression label the STXBP1 knockout. The lower panel, you see the green channel is the GLP. One feature of STXBP1 knockout is they will go through programmed cell death. The red channel is programmed cell death, staining, and the merge channel you can see. A lot of cell is going through the programmed cell death. That indicate our knockout is efficient. The last one is contralateral control. Show you there is no programmed cell death. That means the programmed cell death is induced by our STXBP1 knockout. With this system, we do the staining. I hope you can see the magenta channel is the staining signal. I use arrowhead label the black holes in the staining. That channel can I use? Okay. Those black holes actually is the position of those STXBP1 knockout cell. Indicates the staining is specific to make it more clear. We culture we isolate those knockout cell. We culture it in the dish that is in the lower panel. The insert is the control. You can see every cell has the STXBP1 expression. That is a red signal. And the main panel is knockout. I use arrowhead, want to label it. In those green cells, you cannot see STXBP1 signal. So all these results show our STXBP1 staining is special, is a specific targeted endogenous one. With this staining method, we do the double staining, that is red panel. You can see STXBP1 co-localized with MAP2, the dendrite marker, co-localized with tau. That should be axon marker, but this is a very naive neuron, so it's also everywhere. Also, STXBP1 co-localized with affecting. That means STXBP1 probably is a universal housekeeping protein expressed in the entire cell body. I also want to see during the different developmental stage if STXBP1 is universally expressed. Actually, it is. Even in the early stage, I checked the early stage like E14, P0, all through P28. They all universally express STXBP1. In the later stage, we want to see if when they already develop synapses, if at that stage STXBP1 specially located in synapses. So our senior scientist Roger Banerjee used a centrifuge, isolate synaptosome from cytosol. That is in panel B. We find STXBP1 has a higher concentration in synaptosome, but there is a substantial amount of STXBP1 in cytosol. That means STXBP1 really universally expressed during the entire developmental stage. So our next question is, what's the function? So there is a problem because we knock out STXBP1 directly trigger program cell death. You can see in the upper left panel when the pups just born. You can see the red color. That's we label the neuron. If we knock out STXBP1 at that stage, we still can see the color. But several days later, the color disappeared. They all go through programmed cell death. All the neurons died off. We have the brain slice in the lower panel. So to overcome this difficulty, we try to use a rescue experiment. We can, although we cannot see the knockout defect. We can use a pathogenic mutant rescue the knockout program cell death. And this is the result on the right panel. If we use a wild-type STXBP1 rescue, the pyramidal neuron is the morphology is roughly normal. There is apical dendrite, basal dendrite, everything. If we use a benign STXBP1 rescue, it's not fully rescued, but it's okay. There is still polarity. The cell body, there is a polarity on the cell body. The lower panel shows 3 pathogenic mutation rescued sample. So the polarity, the pathogenic mutation can rescue programmed cell death, but the cell lost their polarity. The cell body is round, and the cell body cannot grow out, cannot extend their processes. So with that, we want to see the molecular level. What's the STXBP1? Oh, sorry. What's the STXBP1 binding protein? What biological processes STXBP1 involved to control this morphological development and control the polarity building? So we use immunoprecipitation and mass spectrum to identify STXBP1. We use antibody pull down all the STXBP1 and its binding protein run through mass spectrum. We identify 114 genes. We upload the list to David database. Annotated, we find these processes. The bar chart is individual biological processes. So as you can see, besides the synapses process, STXBP1 is also involved in, based on the annotation, STXBP1 is also involved in myelin sheath generation, also involved in cytoskeleton F-actin processes. We use mutual immunoprecipitation to confirm this result. We choose alpha-2 spectrin and ARPC. They are all involved in actin. Involved in F-actin assembly. So we found that the binding is true. Those 2 proteins can really bind STXBP1, and we use immunohistochemistry, stain those 2 proteins. We find STXBP1 can control these 2 proteins. With STXBP1, alpha-2 spectrin locate on the surface of the neuron. ARPC also locate on the surface of the neuron. Without STXBP1, you can see in the lower panel, they cannot locate on the surface of the cell. Okay, so because STXBP1 is involved in the F-actin related, is relevant with F-actin related protein. We want to know if STXBP1 actually involved in radial migration. We did spend several years to investigate, to study how radial migration determine the cortical layering and the infrastructure. This is a parallel research. We found in this mutant mice, the cerebral cortex is much thinner than normal. I hope you can see it in the red panel. In early stage, before perinatal stage, the cerebral cortex and the wild type are comparable. But in P5, P7 stage, the upper cortical layers become much thinner. This is exactly the cortical genesis period of upper cortical layers. I want to show you in left panel, those are in perinatal stage. Those migrating neurons, they form vertical lines. I did the time-lapse imaging to show those migrating neurons, how those migrating neurons generate position in the emerging cortical layers. So in the middle panel, in the middle upper panel, I use the green line label the dorsal border of a cortical plate. I hope you can appreciate the neuron that is the top migrating neuron migrate out, bypass the cortical plate dorsal border, and lost their leading process positioned in the emerging cortical layer. I will be quick. The second, the middle panel, I show within the cortical plate. Those neurons form lines. So the leading neuron and the falling neuron, they form a sequential. They have a sequential order. The leading neuron migrate a little bit, then the falling neuron migrate a little bit. They maintain their relative position that means until the leading neuron position in the emerging cortical layer, then the falling neuron will bypass it and position in the more superficial layer. That will add the thickness. The bottom panel show if there is no data sequence, they will migrate together, position in the dorsal border of the cortical plate. They position together, form a dense and a thinner cortical plate. I hope I explained that clearly. And what will STXBP1 do? So as you can see, the wild type, those migrating neurons, they spread in the upper cortical layer in a wide range. And if we knock out STXBP1, they all migrate to the dorsal border. That means that indicate STXBP1 may involve in the directly involved in cortical genesis and may involve in early maturation and early onset those symptoms. Thank you for your attention. Those are keynotes you can take home. And I would like to thank our lab mate, and I would like to thank Dr. Yu Wang, Dr. Roger Banerjee, and Jack Parent for their generous support in STXBP1 project. I also like to thank Professor Roman Giger, Peter First, and Xiaofeng Zhao voluntarily helped me finish the neural architecture, cortical architecture project. Thank you. I would like to take your question.